In this segment, we're going to look at how people have gone about studying the effects of early experience on subsequent behavioral development. So I think it goes without saying that early experience affects later behavior. Maybe not all the time, because there are discontinuities in development, as we've seen in another segment. But, I think more often than not, we're going to see some kind of trace effects, and maybe more than just trace effects, but really huge effects of early experience on the way we behave later on. So basically, let's look at some of these modes of developmental analysis, ways that people have gone about studying the effects of early experience. So we're going to start with something called attenuation. That's our first mode of developmental analysis. So if you are attenuating a particular experience, you're going to be giving less stimulation than would normally be encountered. And so we can draw it out this way on a timeline. In fact, for each of these modes of developmental analysis, we'll use a similar scheme. So here's the one for attenuation. So just as a frame of reference, we have these events the animal is encountering at these different points of time. So at time 1, A, B, and C happens. At time 2, D, E, F, and at time 3, G, H, I. So if you're doing an attenuation type of study, what's going to happen is you're just going to chop some of the experiences out. You're going to take away things that otherwise would normally be encountered. So all that's left in time 1 is A and C, time 2, D, and GHI are gone altogether. And then you'll look at the effects of attenuating these forms of stimulation or motor experiences on subsequent behavioral development. So this is pretty much what Harlow did with his infant rhesus monkeys, that he separated from their mother. So he attenuated maternal experience and instead gave them these cloth surrogates to cling to. But basically, just by removing them from their mothers, he was automatically attenuating the kind of maternal experiences that a young rhesus monkey would otherwise have. And of course, later on, he went on to look at the effects of attenuating that on various types of abnormal behavior patterns, depressive type symptoms, as we see, especially in the photograph over on the right, with a rhesus monkey that's kind of sitting in a corner, all huddled up, and we can't see in a still image, but engaging in stereotyped rocking back and forth movements. And so the idea, of course, was not to create horrible, abnormal monkeys, but to use that as a model to try to deal with finding out ways to actually alleviate depressive symptoms. Because if you're going to alleviate depressive symptoms in a non-human organism, first you have to create a depressed non-human organism. And at the end of this segment, we'll actually look at how that actually ended up panning out. Here's another experiment on attenuation, a classic one by Heldenheim in this gondola apparatus, as it came to be known. And basically, we have two kittens. And one you can see very clearly here in the front, and it's just being restrained, gently being restrained, but it can walk around, and as it walks around, this thing swings around, so it's just kind of walking around this circular arena. What you might have trouble seeing is this kitten, there's the head of the kitten, that's being passively carried in this box, and that it's sort of like a gondola, okay? That's why this came to be known as the gondola apparatus. And so every step this kitten takes, this kitten is being traversed the same distance, but it's not in contact with the substrate. Of course, it's in contact with the bottom of the box, but it cannot control the visual stimulation. It's visual stimulation without the motor component. There's no visual motor coordination here. This one is visual motor. It moves and it can see changes in this wall's pattern, and the wall was painted intentionally this way to maximize the amount of visual stimulation that the kittens received as they moved around the wall. So did attenuating the motoric component of visual motor stimulation matter or not for the gondola kitten. And it turns out that it did. So Heldenheim gave a whole variety of very simple tests, and this is just one, a paw placement test, where you gently lower a kitten to a table that has these prongs sticking out. And basically what happens is the one that had visual motor experience, not the one that was in the gondola, was able to actually reach out with its paw and hit the prong. But the one that was in the gondola had a great deal of difficulty doing that. Its paw would go in between. It had a really tough time coordinating the movement of its paw with the visual object of the prong sticking out right in front of it. And they also used a visual cliff, and they found that the gondola kittens, the ones that were being passively carried around, were more likely to walk over the deep side. Again, they're on a glass surface, they're not going to fall, but they weren't perceiving depth very well. And of course, the ones that were in visual motor control avoided the deep side. Now, interestingly, and this is a side note, but I think it's an important one, they actually looked at these kittens a year later. So now they're cats, right? And what they found is that the passive ones, in other words, the ones that were in the gondola that were passively being carried around the apparatus, now were like those active ones, the ones that had visual motor control right from the get-go. They were more like the active ones were about a year earlier. They basically caught up. It took them longer to do it. So now, a year later, they could do the paw placement test, they could do the visual cliff, and the other various types of tests that they administered to the kittens and re-administered to now the cats. So they ended up developing ultimately fine, but it took them longer to do it. So in another segment, we looked at 4 roles of experience. Okay? So which role does this clearly illustrate? Hopefully you're thinking facilitation, because that's exactly what was going on here. So there was a deficit, but it only mattered in the short term. They eventually caught up. How to eat a mealworm. So this also revisits another example that I used in a different segment, but I'm using it here in the context of an attenuation study. So here we have an adult bird that is handling a mealworm perfectly, grasping it with its foot, and then nibbling it, putting it in its beak without dropping it, tearing it apart. Poor mealworm. Holding it, doesn't drop it, is able to handle it quite well. So that will hopefully bring to mind this study, the Wallman study, with the chicks that were outfitted with booties so that they could not see their feet. And you'll remember that by attenuating that visual experience, the chicks that could not see their feet because they had these dark booties on had a difficult time handling the mealworms. They had a difficult time picking them up, but even if they picked them up, they would drop them, and they really could not eat them very easily. So attenuating the experience of seeing your feet, if you're a chicken, affects in a negative way handling mealworms. Okay, the second mode of developmental analysis is called enhancement. It's actually quite the opposite of attenuation. So instead of the normal amount of stimulation that an animal would normally encounter early in life, now you're going to be giving it more than it would normally encounter early on. And so if we look at how we would draw that out, it would be something like this. So there's our frame of reference, which remains pretty constant here. And then an enhancement situation would be adding more relative to the normal situation. So notice there's more letters being filled in here, and even in between times 1 and 2 and 2 and 3. More experience than normal. Enhancement. So here's another old example you've seen elsewhere. If you shine a light in an incubator, well, the eggs will hatch sooner. But of course, normally eggs are not hatched in the light. They're underneath of a hen, or in an incubator, they're in the dark. So you are giving the developing embryos more light, more stimulation, in other words, via light, than they would normally encounter and it affects their development. It speeds it up. What about early diet? Does that matter in terms of what you eat later on? Turns out, if you're a rat, it does. So in this study, they gave rats at a young age a varied diet, more than the kinds of things they might normally encounter, unless they're in an alley going through dumpsters outside of restaurants. They might get that kind of a varied diet. Who knows? But they gave them a little bit of anything that they would take, anything that they found edible, they gave these rats when they were young. And by doing that, later on, they were much more likely to accept strange, novel foods, not a Big Mac, but things that they had never encountered before, than would rats that had a just normal rat diet, the kind of things like that rats would normally eat. I mean, if they're lab rats, it would just typically be Purina rat chow pellets or the equivalent. So by varying the diet early on, you are now increasing the likelihood that they will accept new things. They'll try new foods. A good thing for parents to think about with their little kids. Here's another one, handling. If you handle a rat early in life, just pick it up and pet it. Okay, just like that. So you're giving it more stimulation than it might receive if it's just in the cage without being handled. You actually can reduce stress at a later age. They are much less stressed out than our unhandled rats. This is a really neat experiment, I think. It incorporates a lot of different things, even attenuation, as we'll see, but I'm going to try to emphasize more the enhancement aspect of it. So these are rats, and I think I may have used this in an earlier segment, I'm not sure, but rats that were reared in a variety of conditions. So we have the standard lab situation, 2 rats in an average size cage, and then we have the impoverished situation, a smaller cage, only 1 rat in that cage. They're in a room with other rats, but they can't see them. They could only smell them and hear them. And there was, of course, no social contact either for the impoverished rats. That, by the way, would be the attenuation situation here. But the enhanced situation is a larger cage with lots of rats to play with, lots of toys to play with, and the toys get changed periodically so they don't habituate to them. So for a lab situation, that is definitely a form of enhancement, this enriched environment. So what they found as a result of doing that is relative to the standard situation, the enhanced rats had many more dendritic spines in the brain. And interestingly, the effect was greatest in the visual cortex, the occipital lobe, which is something that caused them to do something else that we'll see in a moment. Okay, so many more dendritic spines, meaning many more connections going on in the brain than would normally be the case. So what we're looking at here is just a summary of a whole variety of things that they found, including behavioral tests. So I'm just summarizing without choosing any particular test. On every metric that they looked at, whether it be dendritic spines or behavior or whatever, they found that the impoverished rats did poorer than the standard rats. No big surprise there. That was attenuation. But the enriched rats did better than either other group. Well, certainly better than the impoverished, but also better than the standard rats. And I guess that wasn't overly surprising either. But going back to the visual cortex kind of thing and the dendritic spines, they wanted to see whether or not it was just the visual stimulation of being in an enriched environment, or you had to actively interact within that environment with the other rats and toys. So they created this observer condition where they had the big enriched environment, but they put small cages inside the enriched environment with 1 rat per cage. So, they could see the other rats, but they couldn't really physically interact except through the wires, I guess, with their paws or sniff one another. But they couldn't interact among the other rats that were not in these small cages within the observer condition. And sure enough, what they found was a significant deficit on all of these tests that they gave the rats, including the occipital dendritic spines. So they were more like impoverished rats. So not only do you have to be in an enriched environment, you have to interact with that environment in meaningful ways, meaning with objects in the environment, toys to play with, etc., as well as other members of your species. But then, and this is the one that I really like, and this is a little known finding. They did not go out of their way to really emphasize this, but I found it. It's what they called the semi-natural habitat. Everything we've seen so far involves cages of varying types. So they had this huge outdoor enclosure. It was all fenced in, and there was a wire on the roof so the rats couldn't jump out or anything. And there was sand, and they could burrow. They could do what rats do in the wild. And there were a whole bunch of rats in here. And so what they ended up finding with this is that the semi-natural rats were better on everything, even more so than the enriched rats, which is kind of interesting because enriched in a lab is certainly relative to the standard lab situation. But in a way, the enriched condition in the lab is just another type of attenuation or impoverished situation relative to what goes on in the real world, which I think is a real important finding, and too bad it was underemphasized. Okay, our third mode of developmental analysis is substitution. And so here you're taking away one form of stimulation that would normally occur, but you're replacing it with something else that wouldn't. So you're neither attenuating, really not a lot. I mean, there is an element of attenuation here. And you're not enhancing either. You're just replacing. Okay, you've taken away something that they would normally encounter and now putting something else in its place. And so on our timeline, we could look at it this way. Instead of A, B, and C in time one, it's N, O, and P, and so on down the line. So they're taking away what's normally there, replacing it with other things. Well, it goes right back to Harlow. So although I talked about Harlow earlier as a form of attenuation, because, yeah, the monkeys were denied experience with their moms, they were, in fact, given a substitute form of experience, the surrogate mother. So the Harlow situation is an example of both attenuation and, in this case, substitution, because here I'm emphasizing the fact that it was given something else instead of its mother, a cloth surrogate. Imprinting, of course, is a classic example. So you're taking away a conspecific that they would normally be raised with, but you're replacing it with an imprinting object of some sort, whether it be another species altogether, as we see in these examples, or some artificial object. So imprinting studies definitely fit this category. Or going back to something that I mentioned very early in the course in a different segment about how Xing Yang Kuo reared a rat and a cat together. Normally they would not be friends, but here they are sharing the same milk bowl. So, you're replacing whatever conspecifics they would normally be with together, and instead giving each of these animals one another as a substitute rearing partner. Okay, and let me just mention one human example. It has to do with premature infants. Because normally, if you're a full-term infant, you get a lot of experience in the womb. You're rocked around as mother walks. You're jostled. So there's a lot of movement going on. But if you're premature, you're in an incubator and you're not moving around. I mean, there is none of that kind of experience going on. So some pediatricians decided to create a rocking waterbed that gave the premature infants the same kind of gentle rocking type motions that they would get if they were still inside the mother's womb. And they found that it actually enhanced a variety of things that they were measuring, that they would typically measure in a newborn, various reflexive type responses and all kinds of things that obstetricians do when an infant is born. So they actually gained, as a result of being on these rocking water beds, instead of just lying in a regular old incubator. Okay, the fourth mode of developmental analysis is called transposition. Now, this is a strange one because there's not too many examples that fit this, but I came upon some that forced me to create this category. These modes of developmental analysis are from a book chapter that I wrote, so I kind of made them up. But that's neither here nor there. So here, you are neither attenuating nor enhancing nor substituting anything. You are simply taking things the organism would typically encounter and rearranging them in some way. And you'll see what I mean by the example I'm going to talk about. So there's our frame of reference. So instead of ABC, it's CAB. Instead of DEF, it's FED. Okay, we'll leave GHI alone. Leave that one alone. Okay. The example is a really interesting one, I think. It involves indigo buntings that migrate at night. They're night migrants. So they rely a lot on constellations, as we see here. Okay, but what is it about the constellations that they rely on? By the way, they can rely on other things as well. Who knows? They may have some kind of a magnetic sense. I'm not even sure that's been looked at in the indigo bunting. But, you know, the constellations, if they're up there, they're available for the animals to use while they're migrating. But what is it about them that they're actually using? So this is an observatory where the study was done. It was actually done at Cornell, I think. Not I think, I know. Yeah, because I know the guy who did this work, Stephen Emlen. And what he did was to rearrange the sky, rearrange the constellations in a very clever way. First, he devised these cones. And other people have used cones like that. Since then, we even saw one in a video clip in another segment. A little bit different than what Emlen did, but Emlen's, I thought, were quite clever, because at the bottom of each cone, he had an ink pad, of course, on the inside. And then he used like coffee filter paper, that type of paper, to line the sides so that, and there's a screen, of course, on top, so the bird can't fly out, but it would try to jump in the direction that it was trying to migrate toward. But it would fall back, it would slide back and get its feet in the ink, and then every time it jumped up, it would leave tracings of its footmarks on the filter paper. Very clever. So there's our ink pad right there and the filter paper. And you can see the marks on the filter paper that the bird is leaving. And just by looking at the darkness of the marks, you can see if the bird is showing any kind of directionality in terms of where it wants to go. And this bird definitely is. So here's what Emlen found. Those are some of the actual pads right there. So you can see that we have varying degrees of orientation or not. He also ultimately converted these to vectors so he could do some statistical analyses on them, but you know, we're not going to get into that level of detail here. I think you can see very clearly this bird is not oriented at all in any particular way. It's all over the place. This one wants to go north, this one wants to go south. So there's the planetarium. Okay, the stars, the constellations, that's what I'm trying to say. So normally the constellations rotate around Polaris, the North Star. So he had a condition where he kept that, okay? But then he had another condition where he switched things around so that instead of rotating around the North Star, they were actually rotating around the star Betelgeuse. And so that was now the axis of orientation. Normally, the North Star would be the axis of orientation. So he's basically reconfigured, or, as I called it, transposed the sky. But all of the elements are still there. All the constellations are still there. It's just that they're now moving around Betelgeuse instead of around the North Star. So, there's our indigo bunting. There's the control bird. There's no stars. It's a dark sky. And again, this is all done inside of the planetarium because he needed to control the axis of orientation. So there's the North Star and there's Betelgeuse. Okay, here's the results. So if there's no stars, the birds don't know where they're going. And, you know, there's really no magnetic sense kicking in here because they're in a planetarium. In the wild, they would probably start to resort to other senses, but they're indoors. So they're all over the place. With the North Star, they are heading in the correct direction. They're heading south. They're looking at the North Star. That's the point that they're orienting toward, looking at the constellations moving around it and flying south for the winter. If he switches things to Betelgeuse as the axis of orientation, they are now still moving south. Meaning, it's not the configuration of the constellations per se, because they've all been switched around due to the different axis of orientation. But they're looking at the axis of orientation. Where's the star that they're moving around? Look for that star and then fly south from there. And that's exactly what they're doing. They're not learning, well, the Big Dipper should be over here and Orion should be over there and there's the... No. It's what are these constellations moving around? It's that axis of orientation that is important to the birds. Fly south from that point. And that's exactly what they're doing here. The final mode of developmental analysis is called displacement. So what you're doing here, it's basically a critical period or sensitive period type study. You are just shifting temporally the stimulation. So something that would be encountered at one point in time isn't encountered then, but is encountered either earlier or later. Like I said, a typical critical period type study. So relative to our frame of reference, A, B, and C is now being encountered at a later time than at its normal time. This brings us full circle to talk about Harlow's work with these depressed monkeys. So early in life, they are denied not only maternal experience, but also peer experience. They are in isolation from other monkeys, and that induces depression. And depending on how severe the isolation is, because he used different types of severity there, you could end up with an animal that is basically showing the same kind of clinical depressive symptoms as a human being does who is severely depressed. And in fact, the monkey on the left is one of those monkeys. So they didn't have peer or maternal experience early on. So he gave them peer experience later on. From other work we know peers can be more important than even mom. So these were monkeys. These he called them therapist monkeys, the healthy ones, the ones that were raised normally with mom and their other siblings. They were trained to approach these other monkeys, the depressed ones, and try to interact with them, and we see that happening right here. So now the depressed monkey is getting the kind of social experience that it should have gotten at an earlier age. Now it's really severely depressed. And what Harlow found, along with his colleagues Steve Sumi and Melissa Novak, is that they weren't totally cured of depression because these were severely depressed monkeys, but they really became significantly better instead of having not had this form of therapist monkey experience. So there's our client monkey up on top that is initiating play with the therapist. See, now it's the depressed monkey that's actually doing the initiation of the play. It is not totally come out of that depressed state, but it is a lot better than it was before. The severity of the depression has been alleviated using this kind of a paradigm, displacement. So those are our modes of developmental analysis: attenuation, enhancement, substitution, transposition, and finally, displacement. So that takes care of not only all of the development segments of the course, but all segments of our animal behavior course. I hope that this has been a beneficial experience to the students who have experienced this.